Hello and welcome to the Randox webcast, COVID-19, the full diagnostic circle. My name is Chris Chambers and I have alongside me here Dr. Dwayne Vance. So before we get stuck into today's content, I would just like to take a quick moment just to praise the efforts of all the clinical laboratories all over the world who are currently aiding in the good fight against COVID-19. The work you're all doing is extremely important in helping to diagnose and basically mitigate the spread of this what's been a terribly horrible disease that's been among us for way too long now. But we all hope with the news of all the vaccines that are getting rolled out all over the world that we hope that things will get back to normal sooner rather than later. Okay, so as I say, we've got Dr. Dwayne Vance here, and in 2020, last year, Randox were involved in the creation of what's now is basically a COVID-19 super lab. The lab was built with the intention of assisting the UK government in the fight against COVID-19. And we have Dr. Dwayne Background here, as I say, who was a former lab manager of that facility, and Dwayne is just going to take us through, basically in terms of the job that he did while he was the manager in the lab, and then what we're going to do is we're going to basically talk about the full diagnostic circle. So beyond COVID-19, the diagnosis and basically what really should be in store or what is in store for diagnostics moving forward. So I'm just going to call you Dwayne for the purpose of this today.
No problem whatsoever, Chris.
You obviously don't mind because we know each other very well. So Dwayne, can you take us through, just give us a little bit of a brief synopsis about your time. We're just trying to speak in general, really, about labs in general. But if you could provide us with a little bit more context of your time running what is arguably Europe's largest standalone COVID-19 testing facility.
Yes, of course, Chris. It would be my pleasure to give you a brief insight to how Randox initiated such a fantastic super lab, but also to give you more of a bit of a personal aspect of how my time, how I see it in the front line of diagnostic testing for me took place throughout the year. So way back in February, which seems such a long time ago now, we started reacting to the global crisis of COVID-19. Our first instance was to really look at how we could get involved with diagnosing COVID-19 by developing our own PCR assay using some of our own biotechnology, such as the Randox biochip that some people may have already previously heard about. So quite quickly, in fact, within a period of 2 weeks, we had developed one of the world's first PCR assays and went on again then to get it CE marked. And this was one of the assays which we actually routinely use within the COVID labs at Randox laboratories. I remember becoming involved within the COVID lab. Back then, I was working as a molecular product specialist. And although I had left the laboratory setting, one Sunday evening, I received a phone call asking me if I would like to go back into the lab to act as an extraction scientist. And of course, I myself thought this was a great opportunity to get back into the lab and to get back on hands-on again with doing extractions. And back at that stage, we were processing between 80 to 130 tests a day, which seemed back then like a large number. Mm-hmm. But little did I know, 10 months down the line, we'd be doing 100,000 per day. So, and you can imagine the infrastructure that took. A lot of people were involved in laboratory design, infrastructure, purchasing, finance, recruitment of staff, redeployment of current staff into the labs, such as myself. So one of the purposes of this presentation, this educational workshop here today, is to really give an insight and an overview of how to establish a COVID-19 laboratory. So designing a molecular lab is something that's happened globally now over the last I guess 12 months. But Randox was lucky in that regard, because we had our own accredited molecular lab in Randox for our own tests, such as sexually transmitted infections, influenza, influenza A, influenza B, and our own respiratory panel. Of course, that didn't include SARS-CoV-2, but included other coronaviruses. So we had the infrastructure, albeit at a smaller scale. So we were able to look at our infrastructure and really expand and magnify, or pardon the pun, amplify the technology quite well. So
well, I have there from time to time myself.
Yes, that's true.
Obviously, nowhere near to the degree that you did, but you know how quickly the place grew. You know, from basically what was, as you have already said, a very small amount of tests, into what you've said now is, you know, became a huge amount of tests. Is, you know, it's highly impressive. You know, the transition that the place made over such a very short period of time.
Yes. So it was quite a simple process then to really expand our current facilities. So bearing in mind that the technologies that are mostly used within a molecular setup are based on a traditional workflow. So a traditional molecular workflow follows a unilateral one-way system, and that's just to ensure that there's no crossover contamination between different chains within the laboratory workflow. So, for example, when the sample comes in, which is known as your accessioning department, that's where the sample comes in. The samples are logged onto the LIMS system. The samples are decontaminated because we're not involved in the transportation of the samples. We need to ensure that the samples are safe for handling. So they're decontaminated and they're scanned into the LIMS network, or the laboratory management system that may be installed within the lab. Samples are then batched into an appropriate batch number. So the majority of molecular labs for COVID testing would be based on 96 well plates, or perhaps 384 well plates. So once samples are batched and logged onto the system, then they can be ready for sample processing. And the first part of the molecular process is your extraction facility, and this is what I refer to as the powerhouse of testing, because that's where the samples are being brought in their live format. So the extraction lab will require special equipment such as BSL level 2 laminar flow cabinets.
No doubt this is where you spent the most of your time then.
Yes, I was very much involved in the initiation of the extraction process. However, as a laboratory manager, you are responsible for the entire process, from accessioning to extraction to PCR reagent testing, as well as the real-time PCR and the Randox biochip detection department as well. So once their samples are extracted, either using manual methods or automated systems, we were very lucky to have multiple different forms of automated extraction platforms available to us, which meant that we were able to increase our capacity and performance quite well. But there are so many different variations of extraction systems. You can have manual systems such as spin columns, which would be more suited to the smaller laboratory setup. You will have cartridge-based extraction systems, which would be very much suited to those labs operating at a medium capacity. And then you have these fully automated rotational-based systems that would be very much suitable for those labs that are expecting very high throughput, such as the labs that we had at Randox. So once the samples have been extracted and purified of their nucleic acid, in this instance, the single-strand RNA of the coronavirus, the samples were then transferred then to either short-term storage or else to the template addition laboratory. And that's where the RNA from the extracted material would be added to the PCR reagents. So just bear in mind, whenever you're preparing your master mix reagents, It's vitally important that your master mixes laboratory is located as far away as possible to the amplification detection room to prevent any carryover. Your master mix laboratory must remain biological free. No biological material should ever enter the master mix room, and that's to ensure that the chemical reagents that comprise of the PCR reagents are kept clean and in a sterile environment, and that's just to reduce any contamination that may be evident within the molecular lab. So it's all about damage mitigation and ensuring that you've included quality management and quality processes in your lab to reduce contamination to as much as possible. So once the RNA is added to your master mix PCR plate, then that's where a seal is added and is then put through then for real-time PCR amplification or endpoint PCR, whichever technology you're using. And the PCRs typically last for about 1 hour to 2 hours, depending on the assay that you're looking at or the targets that you're looking at. And you will either receive a result on the patient diagnosis during the amplification process in the term of the real time, or at the end of the process of the PCR, such as endpoint. And if you are using endpoint analysis, then you will have a subsequent department within your lab that will be responsible for the detection. So, for example, within the Randox lab, not only did we use real-time PCR, but we used biochip array technology, which is Randox technology. It's got the capability of undertaking a step called DNA hybridization and conjugation, which can result in the qualitative detection of the coronavirus gene targets by producing a chemiluminescent signal, which is spotted under a special type of camera within the Evidence Investigator platform. And alternatively, smaller labs may use other technologies such as gel electrophoresis, capillary sequencing, and then some very advanced laboratories will also use some form of viral genome sequencing, such as next generation sequencing as well. So there's a whole host of different technologies and systems that can be incorporated within a molecular lab workflow, but what they will all have in common will be that they follow a traditional molecular one-way unilateral flow work layout.
It's great you had so many different options at your disposal there whenever you guys were carrying out the tests within the lab, so you weren't just confined to one method of testing essentially.
No, and that was very important, especially during a global crisis. There would be instances where there would have been shortages of a reagent, an extraction reagent, or a shortage of a PCR reagent. Because we were lucky, we had our own certifiable extraction systems and real-time PCR reagents and evidence investigators. So if there was ever a shortage of real-time reagents, then we always had multiple different workflows within the lab, may it be biochip or real-time PCR, and as well as using multiple different types of extraction. So and actually over the last few months, it's the opportunity of getting involved in COVID-19 has given us a real drive to even develop our own reagents. So we've also developed our own nucleic acid extraction kit, as well as our own real-time qPCR reagents as well. So again, it increases the portfolio and the options that the end user may have whenever they go to continue their COVID-19 diagnostic testing.
You probably didn't think about it at the time, but excuse me, looking back on that time, you probably just jumped in, I would imagine, and just got stuck into the work. But looking back and reflecting on that time, you must be pretty proud of yourself, because that's a fairly substantial undertaking that you've been doing, and for all of society, basically. How did that feel for you personally?
Well, as a molecular biologist, I found it pretty exciting. Of course, there was so many unfortunate and sad news events that were being broadcast around the world at that time. But as a scientist, I was very much putting my hands in and getting them dirty with regards to getting as much and trying to ensure the health of a nation. Obviously we're based in the UK, so it was getting involved with the UK diagnostic side of things, but I could just imagine all around the world, people in my shoes were doing exactly the same thing. So it was nice to feel a part of like an international effort to try to curb the really bad global pandemic that was happening right in front of our eyes.
Yes. So what you train for while you were a student? You probably never realised that we would ever be in a situation like what we came to be in. But luckily you had the skills and knowledge to be able to apply that to a very practical level, and what's turned out to be a really great effort. So that's obviously great. Okay, so you touched briefly on the different tools that were at your disposal within the lab. What about the actual COVID test? What's very prevalent at the moment, Dwayne, is you see in the news. I know you're not a big reader of newspapers, but I read a lot of newspapers day in, day out. I also watch the news occasionally, but what I can see is very prevalent is all these different mutations. There's there's kind of different mutations that are getting named on the news at the moment. So how does the assays that you're working with within the lab, is there any implications for those assays, or how do they stack up against these new variants?
Yes, well, back in February, March time, just after the viral genome of the coronavirus was made public by the Chinese government, the multiple diagnostic companies such as ourselves went straight online to look at the viral genome. Let's look at the genetic makeup of the virus to see exactly what the genetic sequence is, and let's look at designing little chemical tags called primers and probes to see how we can accurately detect this virus in human samples, such as nasopharyngeal swabs. So there was multiple different genes identified and open reading frames identified within the viral genome of SARS-CoV-2. So, for example, you had your spike protein, your N protein, your E gene, as well as your ORF genomic regions as well. So each diagnostic company really came to design their own specific test. Randox undertook a lot of some time and ensured that we identified areas that were conserved, specific, but also ones that would future-proof the assay. So specifically, we looked at the E gene, which is a test that looks for all different beta coronaviruses under the subgenus Sarbecovirus. So any SARS-related virus, SARS-CoV specifically, as well as SARS-CoV-1. And we also looked at open reading frame 1AB, which is specific uniquely to the SARS-CoV-2 virus. Other manufacturers looked at the E gene and the S gene, and this is where the mutations really cause some issues for some assays. So some of the assays out there have looked at the S gene, and because of the mutations within the S gene, then that's caused some issues with sensitivity. So you may find now that some laboratories that were using assays that looked at the S gene may now have converted to a different assay, or they will ignore the result of that particular gene target. Or if a patient is positive only for the S gene, then that patient might either be rendered a negative, or might initiate a repeat of that patient's swab, or may request a new sample for that particular patient due to the issues of the S gene causing sensitivity issues for those assays that have it. So you will still see a few on the market, but the majority of those now have developed version 2s where they look at the N gene or the E gene or the ORF genomic region.
In essence, the Randox test is totally unaffected by it then?
Yeah, we're very lucky that we've got such a good, strong team of research and development scientists that are able to predict this, and that was their main reason for choosing the E gene and the ORF gene as well. Okay.
All right. Okay. Just on the instruments again. So you're what you were saying. Your lab is traditionally made up of essentially split up into extraction, PCR, and detection. That's how the lab operated. But in the future, we're excuse me, there's a lot of all-in-one testing solutions coming on the market. Where do you see the future for these type of devices?
Oh, I think there's such huge potential for point of care testing, or you may also be referred to as near patient testing for COVID.
So by all-in-one, what we mean is that all of the extraction, the detection, extraction, amplification, and detection is all contained within a single unit.
Yeah, and that is really the crux of point of care. So the idea is that a sample is inserted into a cartridge-based device, and that cartridge is then inserted into a very small portable detection suite about the size of an espresso coffee machine. At Randox, we're actually involved in the development of point of care cartridges for a system called the Vivalytic. And it's your quintessential point of care system. Sample is inserted into an external cartridge by Randox, and then the cartridge can terminate. And so we've worked very closely with this engineering system, this engineering company called Bosch, to provide a really sensitive platform for COVID-19. Minimal training, no scientific background or experience is required for actually using the machine. So after 30 to 45 minutes, depending on the cartridge that has been used, it produces a diagnostic report. And depending on the test that the clinician has requested, it can either be a qualitative PCR test, or it can be a quantitative PCR test, which would provide you amplification curves as well as a CQ or CP value, which is indicative then of the level of infection that patient might have.
At the point of care, that's really impressive.
The technology and the technology within the molecular diagnostics field at an exponential rate, and it's exciting to see treatment and testing being carried out potentially at the bedside, but also in airports. People can travel if they know they've got a negative test result within 30 minutes of boarding their flight. So again, it makes things very accessible to the majority of people now.
Beyond the point of care, we see a lot of all-in-one solutions coming on the market. For maybe sort of the more kind of medium throughput labs. Where do you see that type of technology moving?
Yeah, well, again, COVID has kind of really brought these all-in-one systems to the fore. Over the course of the last 10 months, you'll have systems that are based for centralized laboratories, not specifically molecular centralized laboratories, but general clinical chemistry, immunoassay serology labs that don't require that traditional molecular workflow. Because the beauty of these all-in-one systems is that everything happens in a concealed environment. So you've got your point of care systems about the size of a coffee machine, and then you have these larger systems that are much more capable of multiple results at the same time. An example of that you should see on the screen at the bottom of the page is the Randox Discovery system, which can analyze many different samples at the same time within one machine. So you've got your extraction in one particular part of the machine, and then in the middle part of the machine you've got your amplification part, and then on the other side of the machine you've got your detection part of the molecular process. And the results can be, results can vary on the type of assay you're using, but you can typically get a batch of 16 results within a couple of hours, and then for every hour after that you'll receive another batch of 16 results. So again, it's perfect for medium-sized hospitals that are maybe have got an intensive care unit where a result is wanted pretty quickly to determine the infection status of a patient.
The great thing about those type of machines, either if it's point of care or the larger machines that you mentioned, essentially the lab staff can get a little bit of pre-work done beforehand, and let the machine go to work, and then they're freed up to go and look at other tasks maybe that they're that are maybe have to be taken care of within the lab as well. So then they can basically wait until the full cycle is done, and it's just a matter of retrieving the results back off the system then.
Yeah, because it's an automated system, it gives you that walk away time, which means you can continue doing administration or other parts of the process, like for example looking up quality assessment data that you might have in your lab, or some other form of ensuring good quality management within your lab as well.
Okay, well that's a good bit of information about the tools that you were using. In terms of running a lab, running any kind of a lab, regardless of being a super lab like the one that you've been working in. Quality control is obviously imperative. Be that inbuilt controls that you get with your reagents, or third-party external quality controls. Can you give us a little bit of a walkthrough about your experience with IQC whenever you were in the day-to-day of running of the lab?
Quality is first and foremost the most important thing within a molecular laboratory. That is definitely, in my opinion. Of course, whenever you're working with a global pandemic in a super lab, quality and quantity is important. So you need to ensure that you can get the desired quantity of results within a specified period, but you also must ensure that your quality is maintained at the very highest level. So in any molecular lab around the world, there will be high-end quality management systems in place to ensure that the quality of your results are as high as possible. And so a lot of PCR assays have built-in controls, which are great because it ensures that the sample is appropriate, and that your sample has been extracted correctly and it's been PCR'd correctly. But also now the international standards are now going a bit above and beyond first-party controls. They're looking now at third-party controls, and not only third-party controls, but controls at our whole genome. So as COVID has went on, in the initial part of a lot of this early controls were partial genome or DNA plasmid controls with only part of the genome in it. Mm-hmm. But now what's happening is that a lot of the virus has been cultured, and some manufacturers around the globe have now started to promote and develop their own whole pathogen controls. Within our own laboratories here at Randox, we use a UK brand called Diagnostics, mm-hmm. Which are whole pathogen controls, which we find are very important, because whole pathogen controls can be used within any molecular workflow. So any extraction platform, any PCR reagents can be used, because you're looking at any target genome, and any real-time PCR device can be used. So you really have that universal applicability to using the whole pathogen controls. So,
talk me through, using of Diagnostics. So where would you have started? Where would you, in terms of your workflow, where would have been the first place where they would have been implemented?
Yes, so at the start, and at the same again would be with clinical chemistry, your linearity, your range, your dynamic range, or in molecular biology we'd refer that to your limit of detection, your LOD and your LOQ. How sensitive is your molecular workflow? What is your sensitivity? What is your limit of detection? How many copies per mil can your workflow actually detect accurately? So whenever we were introducing new extraction platforms within the lab, or looking at different assays, for example, looking at the Randox biochip PCR and then looking at the Randox qPCR reagents, we wanted to look at the LOD for those within our molecular workflow. So what we did before initiating testing of this new technology, we looked at we went onto the Diagnostics and got analytical Q panels. And analytical Q panels are very important to look at your dynamic range of your molecular workflow. So that was very important to look at that.
They're looking at the bottom limit of, say, your clinical range, maybe below that to the range, the lower limit of the machine, and then you're going, you know, beyond that to the upper limits of the clinical range, and then beyond that the upper limits of the machine. Is that what you're telling me?
Yes. So the way that the analytical Q panels have been designed, they've been designed to cover the dynamic range of the assay you're using, which allows laboratory managers to assess the linearity, LOD and LOQ. So that's important because there are multiple different concentrations of samples supplied with your analytical panel. And the reason why the panel consists of a numerous number of samples is to cover that dynamic range of most of the assays that are out in the market in a linear progression. And it's important too as well to note that any diagnostic samples which may be used within a laboratory setting are treated like a patient sample. So they're just being treated under the same circumstances of how a clinical sample would be ran.
So they would behave in the same way as a patient sample?
They would. The material that we have worked with is commutable to a human sample, which essentially just means it's clinically significantly the same as what you expect from a clinical sample. So and then you have your daily controls as well, but the requirements of daily controls would very much be dependent upon the throughput of your lab. So if you're operating a very small COVID testing lab or any other lab that's looking at any other pathogen, you might want to run a Q control within every single run. But if you're looking at 100,000 samples a day, then that might not be economically feasible, but you still need to introduce Q controls in your workflow. So you might want to ensure that there's a minimal amount of Q control going through specific, or at least a number of specified times for each extraction platform. So you're testing the quality of all your extraction platforms.
Okay, so the Q controls would have been your go-to essentially.
Yeah, that would be your go-to, which usually would have been at the medium titer level.
Right, okay. That's fine. And then, you know, like in terms of internal quality control, obviously that wouldn't be the be all and the end all. Whether that be, you know, you said you've used a mixture of the built-in controls that were within your assays, and also the third-party controls. Like, talk us through what other what other feathers to your bow essentially did you have in order to ensure that, you know, that the highest quality of standards within the lab? Yeah,
so a few other examples of how one would use third-party molecular controls would be for lot-to-lot assessment. So as you can imagine, labs throughout the world are using reagents at such high volumes where they may be changing their reagent lot to lot, maybe once a week or once a fortnight, whereas before that, in reality, it could have been once a month or once every 3 months. So the requirements for third-party controls have therefore increased due to the absolute increase in capacity of sample testing. So whenever laboratory managers realize that there's a lot-to-lot change required within their process, then they need to initiate what's called a lot-to-lot assessment procedure. And that's just to ensure that the current lot that lab is using is very much comparable to the new lot that they'll be moving on to. So for that, you might use the addition of patient samples, positive and negative, but it's very important to use your certifiable material because it's stable, it's quantifiable, whereas your patient samples, although they may do what is required, sometimes they can be unstable and cause a variation in results.
Would the internal controls that you were using, the inbuilt controls, would they not pick up those lot-to-lot variations?
Yes, so the inbuilt controls don't necessarily do that. Because it's an inbuilt control has been specifically designed by that particular assay manufacturer. So you need that third party element to identify any lot assessment, and that would be most notified using a Levi Jennings chart as part of your QC protocols.
Okay.
But there are multiple different reasons why you would want to use Q controls. Training of staff, recruitment was such a huge thing within labs, COVID labs around the world.
Because I suppose you can't train staff with live samples, because you can't make a mistake with something like that, can you?
Yes, you can't. They can't be live samples. So laboratory COVID labs around the world really developed some very good training schemes for their new staff, their newly qualified staff. And as part of that scheme, it would be to train staff using negative controls, but also positive coronavirus controls. And that was just to ensure that the scientists at the beginning of their training stages were pipetting accurately, but were also understood the process of how to extract effectively and accurately, just to ensure that the results provided to the patient at the end of the day was as accurate and as high quality as possible.
Okay, so additionally with the third-party controls, all accredited labs also must use external quality assurance. I think you've used multiple, Dwayne, from having conversations with you. Can you talk me through your experiences with using external quality assurance within the molecular lab, in addition to the internal quality, the internal quality control process you have, be that inbuilt controls or third-party controls?
Yes, again, very much like third-party controls, which are required per ISO regulations. The participation in external quality assessment is also very important. There are multiple different service providers out there that can provide you blind samples manufactured to a very high standard. Within our own laboratories, we actually took part in multiple different EQA schemes at variable frequencies throughout the year to ensure that our processes were as watertight and as accurate as possible. One such program which we are using, is again based in the United Kingdom, and some of you may have already heard about this program, or perhaps some of you already are using it within your molecular labs. It's a company called QCMD. They're based in Scotland, and they're a very well-known molecular EQA service provider who provide such a wide and various number of virus and bacterial EQA programs. In fact, I believe their first pilot EQA for coronavirus was one of the first to be made public as a service for international labs around the world. So it was great to be part of that peer review group.
So they would have, so talk me through the process. So I gather you somehow you sign up with them, or you register with them, and then what, you get a batch of samples. You say they're blind, you don't know what they are, or how does the whole process work?
Yes, so essentially what happens is that COVID-19 testing laboratories would be provided with a series of samples, and they're very much designed to resemble clinically significant samples. So the laboratory is required to test these samples using a laboratory setup, assay method, and their particular procedure. The results from the laboratory testing are reported back to EQA, or in this instance QCMD, and within 2 to 4 weeks, or dependent upon the service provider which the lab is using, they'll be provided with an individual report. And within that individual report, there will be details of the overall performance of the participants within the same peer group, but also for that particular lab by itself. And that's just to ensure that their workflow is of high quality. And if there are issues with quality, then the workflow can be adjusted and amended and upgraded as necessary. And again, that's just to improve the overall quality of the results that are being sent out to the patients.
So I assume these aren't actual patient samples, are they?
No, the EQA samples are manufactured within the QCMD laboratories. So the materials that are used within QCMD are manufactured to high quality, and what that means is that they conform to the ISO 13485 requirements. So whenever you're using EQA material, you really need that assurance that the quality is going to be good, so that your workflow is getting the most robust service possible to ensure that your samples are also robust.
And the virus is live or not?
The virus is whole pathogen, but it's not a live virus. They have been heat inactivated or else inactivated by gamma radiation. But it's important to treat them like a live virus because you want to treat them like a clinical sample. So although they may not be infectious or live, you still very much want to treat your EQA samples like a live virus.
Okay, and then so once you get these samples and you would run them, then so with it being external, so obviously what happens then after that? Does someone say you're running a good lab, or what's the next step?
Yes, so about 2 to 4 weeks after you submit your data, all the data is brought together and you get this really detailed and comprehensive report from QCMD, and they'll tell you exactly how you compared with other labs throughout the world that are utilising the same technology as yourself, as the labs that you're using, and you're graded between a certain group, 0, 1, 2, and 3. So 0 means full marks, and then a 3 means that some improvements are required. So dependent on the result of your individual lab, then will determine how much of a process change you need to implement. So I'm glad to say that with regards to ourselves, we've got such good quality. It was fine, but the service of a QCMD report means that they can identify issues if there are any present.
And do you need a specific machine to be able to participate in QCMD, or? No,
all molecular workflows are acceptable. Of course, there are some more popular workflows than others, but even if there's only one, if you're the only person in the world using a particular platform or a particular agent, you can still be included in our general analysis, because within analysis you'll have them all grouped together, and you'll have them grouped in particular manufacturers, and then you'll have them grouped in subheadings as well. So if there is more than a certain number of labs participating, they'll be grouped together in a peer group analysis. But if your technology isn't so commonly used, the results of that would be included under the table.
Right. Okay. So say I was using a machine that a lot of other people use, and I would get my score basically stacked up against other people who are using the same machine, pretty much.
Yes. So during the analysis stage, we'll look at the details that you have sent over. So which extraction platform, which reagent you're using, and that will be grouped into the most applicable slot.
Okay. That's fine. Well, obviously we had a good run through of the lab itself, the setup of the lab, the different instruments that were at your disposal or were at anyone else's disposal. Obviously the steps that were taken in order to ensure that the results were as good as possible. Now, obviously, the end of that chain is obviously providing the results to the patient. But beyond being diagnosed with COVID-19 or not, if people are diagnosed with COVID-19, hopefully their symptoms are not bad, and obviously hopefully they are able to get over that. But in terms of beyond the lab, Dwayne, with all of the different vaccines that we see, again, like I said, I'm an avid reader of newspapers. It seems like every day there's a new vaccine on the horizon. So can you talk me through what are Randox doing in terms of the vaccine schedule that we have going on, or what's happening with Randox in terms of of the global vaccination? Where do they fit in?
Right. Okay. So round docs are primarily involved in medical diagnostics, such as developing diagnostic reagents and controls and things like that. So round docs is not specifically a pharmaceutical company. So we're not directly involved in the creation of vaccinations, but what we are involved in is working alongside key opinion leaders to develop antibody detection systems that would be very much used post natural infection of a virus or post vaccination of a particular pathogen. So most recently, in response to the global pandemic, as more people are starting to be infected, and as vaccination rollout programs are initiated and followed through, there's a real requirement to have antibody detection tests available at a very, very wide scale, at a national scale. So although we're very much involved in the PCR testing, because we're still at that peak of transmission, especially as new variants are identified, it's important as well to look at the surveillance of the coronavirus pandemic, how many people within the population has been infected naturally, and how many of them are required to be vaccinated, and how many people, what percentage of the total population have natural or acquired immunity by vaccination. So again, the vaccination test, the RAMDocs have only just recently released, is a test that very much is unique within the market, and again it involves exactly the same technology that we have previously just discussed with regards to PCR biochip. Okay. The antibody biochip is exactly the same biochip and exactly the same machine, and both of them can be used interchangeably.
So I can see on the screen here it says the SARS-CoV-2 Ig, it's the RBD and the NP. So can you talk me through the difference between the NP and the RBD? So these are the markers that the test is looking for, is that correct?
Yes, so the RAMDocs assay has been specifically designed to detect two antibodies that are responsive to the coronavirus vaccine, to the coronavirus pathogen. The first one is RBD, so that is the spike receptor binding domain, and that's very much going to be antibodies will be initiated once the host has came into contact with the coronavirus, but also if that particular person has been vaccinated using one of the messenger RNA vaccines that have been developed to specifically initiate an immune response for the spike protein. Okay, so I believe you're getting your vaccine today. I'm getting mine tomorrow. So if we were to be tested soon if we were to run our samples through this test, we would flag up positive for the RBD in maybe a week or a couple of weeks' time. Would that be fair to say? So as part of the R&D process, so we have our test has been looked at. So 2 weeks after infection or vaccination, a 100% sensitivity rate was identified post 14 days. So if you've been naturally infected by coronavirus, then you'll be positive for both of those antibodies. If you're just positive for RBD, then it's most likely you've been vaccinated only. But if you're positive for both targets after a vaccine, then it's most likely that you actually had a coronavirus by natural infection, but you may have been asymptomatic and you didn't realise that you had the infection in the first place. So it brings that extra capability of looking at and differentiating between those that have been infected naturally versus those that who have been vaccinated in the last 2 weeks.
So the sweet spot would be, for me personally, would be to flag up both those markers, and that would give you the maximum protection then.
Well, again, the interesting thing about having a new virus within the globe over the last 12 months is because we're still learning so much. So we're still learning about the differences between antibodies developed naturally versus vaccine. We do hear evidence that the vaccine antibodies last longer. But again, we really need to look at that longitudinal data rather than extrapolate predictions. You know, so it's, although there are some very good antibody tests that have been developed, it's going to be very much interesting over the next 6 to 12 months to see how this actually plays out. But it's exciting to see this array here that has been developed by Randox, to see how it can really get involved and significantly at the national level to look at antibody surveillance of COVID-19.
Okay, so traditional antibody tests are basically only looking for one marker, more or less. Would that be the crux of the tests that are out there at the moment?
Yeah, most assays will look for one antibody, but again, because of the Randox biochip, it's a diagnostic grade ceramic chip, which looks for multiple analytes within the same sample. So just like your PCR assay, you're able to look at different targets of COVID at the same time. And you're also able to look at influenza and other coronaviruses at the same time within the one biological sample. The antibody tests for the biochip work on exactly the same premise. It's multiplex immunoassay testing. Okay. So multiple assays, multiple antibodies for multiple targets. So it's not just confined to 2 markers. There's a potential you can go way beyond that. Oh, certainly, and that's very pertinent in the field of coronavirus, because we know there's conditions that are very related to coronavirus, such as cytokine storm syndrome. That basically we have the ability, using that Randox biochip technology, to look at multiple different cytokines within one patient sample. We know that cytokines play a vital role in immune systems, that the evidence is there to support that. So the healthy immune system, cytokines essentially help you fight infections? Yes, you know, cytokines are, you know, they can, yes, but it's the overactive cytokine response that can cause the damage, and that's what's known as the cytokine storm syndrome. So basically, an example of COVID-19, they would essentially throw your cytokines off kilter. The very much would, yes. And in fact, they've become an overactive cytokine response can become inducive to a good patient outcome. Cytokine storms are common, actually quite common complications of SARS-CoV-2 infection. It can trigger viral sepsis. Uncontrolled systemic inflammation may lead to severe respiratory conditions such as pneumonia or even ARDS, respiratory distress, respiratory failure, and multiple organ failure in really severe circumstances that can actually result in death due to cytokine storm syndrome. So if you have a platform such as the Randox Evidence Investigator, you can test for your antibodies, you can test for your cytokines. Up to 12 cytokines can be tested in one biochip for one patient sample, and you can do about 50 samples in about an hour and a half. So it really gives you that capacity to do wide-scale cytokine testing. I listen to Joe Rogan a lot, and he, you know, I listen to him every day on the daily commute to work, and you know, sometimes he's got medical people on, but it almost seems like every single day he's talking about COVID-19 for a period of time, but almost like every second day he has someone on and they're actually talking about cytokines within the system. So, you know, this is obviously very topical discussion at the moment with regards to cytokines. Randox offers a comprehensive range of cytokine testing, and not only cytokines but cytokine receptors as well as growth factors. And these have been specifically selected and designed to assist with COVID-19 risk stratification, monitoring of health and of treatment efficacy, as well as looking at the patient's prognosis and recovery as well. So it's quite important you need to understand the full diagnostic circle when you're looking at COVID-19. So you might start with PCR testing or antigen testing, and then follow that through with vaccination rollout. And then you want to really look at antibody testing and other multiplex testing, such as cytokines. And also another Some other very interesting research has really been discovered in the last few months in response to COVID-19. The kidney disease has become an issue clinically, within the clinical setting, in COVID-19 patients, especially those patients who have underlying comorbidities, or who fall within the over 65 bracket.
Okay.
So it's really important to analyse patients for acute kidney injury. In actual fact, in the United Kingdom, the regulatory body called National Institute for Health and Care Excellence, most commonly known in the UK as NICE, recommended that all COVID-19 patients are to be assessed for AKI, or acute kidney injury, on admission to hospital, as well as having their kidney function monitored throughout their recovery process whilst in hospital. And again, the best way to do kidney assessments from a immunoassay perspective is to get multiple different analytes determined using one biological sample, and that's where the Randox evidence investigator also comes in quite useful. And there are multiple other technologies available as well, but with regards to that, from a Randox perspective, we have developed three multiplex kidney function arrays that can detect renal impairment. So the first panel can look at 7 markers, the second kidney panel can look at 4 markers at the same time, and then the third panel can also look at 4 at the same time. So that gives a total of 14 biomarkers within those 3 panels. And the doctor can then request whatever panel they think is most appropriate for these particular COVID-19 patients. So it's all about really not just it's not just about diagnosing people with COVID-19, but it's really looking at those patients who may be susceptible to a poor prognosis, especially ones with comorbidities. With, for example, the diabetic kidney disease is an issue, and that's where I think there's a lot of interplay with disease. So it's the diagnostic pathway is quite complex with COVID-19, but there are so many good technologies out there and some very good laboratory systems within hospitals around the world that are utilising some very good technology. Some of the technology that we've discussed in this educational seminar today, such as the Randox biochip array technology, can be used for biochip PCR, can be used for antibody testing, can be used for cytokine testing, but also can be used for a kidney assessment. And that really opens up the floor to improving clinical health around the world.
Yeah, definitely. The kidney test is obviously very important. You get to see in the, again, just even in the general news, one of the biggest side effects of that COVID has in the human body is that it causes a lot of organ scar tissue even. So, the fact that we could test to look at things like the kidneys is it's really, really important. Okay, so we're probably coming close to the end of our time here. So that's fine. So Dwayne, thanks a lot really for giving us a good run through of the COVID-19 the full diagnostic circle, talking us through the lab, talking us through the quality procedures and controls, how to run a robust lab, looking even externally in terms of looking outside the lab to things like QCMD in order to maintain your robustness, and then moving beyond the diagnosis of COVID-19, basically moving into the vaccination, and then finally the other tests that are available to look at the impacts of what this virus is actually inflicted on the human race essentially. But do you have any closing remarks?
Yeah, thank you, Chris. Thanks for asking some very good questions, some very important questions, especially as we see ourselves at the peak of COVID diagnosis tests, especially within the UK, considering we are in our 3rd or 4th lockdown now. But yes, I just want to pay thanks to all of my work colleagues not only in Randox UK National Health Testing, but all of the frontline health service throughout the world, as well as the laboratory staff. Sometimes the laboratory staff can be forgotten about as first line health workers because we may not be directly working with patients on the bedside. So I would just like to pay thanks to all of the laboratory staff from all around the world, from the virologists working in the labs, to the microbiologists working in the research and development labs, to all of those people developing new vaccines all around the world. And as well, I'd like to thank all of the Randox scientists as well who are doing some very, very good R&D here as well.
What about the others who helped out from time to time?
And the other people from a non-science background who may have helped with the sessioning, they come in very useful as well. Okay,
that's fine. Well, thanks a lot, everyone, for taking the time to listen to our webcast today. If anybody has any questions for myself or Dwayne, feel free to come to the booth. The two of us will both be there, along with many other Randox staff who will be able to answer any multitude of questions. But if it's about COVID-19, the pandemic, or the molecular testing, thing in general, myself or Dwayne, predominantly Dr. Dwayne Vance, will be able to help you out for sure. So please come along and ask questions, and thanks a lot. All the best.